السلام عليكم ورحمة الله تعالى وبركاته. في سلسلة المحاضرات الخاصة بمقرر الأبلايد فارماكوجنوسي لطلاب المستوى الخامس في كلية الصيدلة. في العرض ده هتاخد بريف introduction عن المقرر بتاع الأبلايد فارماكوجنوسي وأهم المصادر التي يمكن الرجوع إليها أثناء أو خلال دراستنا هذا المعرض أو هذا الكورس. As a starting point for the topic or the introduction of applied pharmacognosy course, we start with definition of pharmacognosy. We need to know what we mean by the terminology pharmacognosy. Actually, there are a number of definitions stated in the textbook. I think one of the famous or one of the comprehensive definition for pharmacognosy is stated in Pharmacognosy magazine in 2019. It defined pharmacognosy as the science of biogenetic or naturally derived drugs, pharmaceuticals, and poisons, and it incorporated various modern analytical techniques to authenticate and quality control of crude drug as well as purified extracts, fractions, and components, and even medicinal foods. From the definition, we know that the science of pharmacognosy involves studying natural compounds, including drugs, pharmaceuticals, and poisons. Study of what? Of natural drugs. Study of their physical, chemical, biochemical, and biological properties. Besides the study of the natural drugs, also pharmacognosy in its definition involve modern analytical techniques used for authentication and quality control of crude drugs as well as their purified extracts, fractions, components, and medicinal or what we call functional food. As predicted from the title of the course, Applied Pharmacognosy, we focus in this course on the methods or the techniques or the tools used in analysis or authentication of medicinal plant and their extracts or their products. In this slide, it summarizes the methods used in analysis of medicinal plants, starting from collection of data concerning an ethnobotanical or pharmaceutical or biological and medical uses, which is known as background. The botany section involves collection and preparation of herbarium sheets, including other uh, techniques we studied in botany or in pharmacognosy, including the good collection practices, the good drying practices, and post harvesting processes. Also, the another tools or the another methods are phytochemical methods or phytochemistry methods in phytochemistry section, including screening of secondary metabolites and screening of their biological activity. As we know, phytochemical studies for can be carried out for fractions, isolates, or even the crude extracts. Another step or another method for analysis of plant is biological testing or pharmacological studies, which involve pharmacological and toxicological studies besides microbiological studies. In microbiology studies, we test the plant samples in vitro, determine their activity against bacteria or fungi or even the virus. And in pharmacology and toxicology, we test the safety of the sample beside their effectiveness in treating or in affecting some conditions or some ailment. After establishing pharmacological studies and microbiological studies, we can go further steps in pharmaceutical studies, which involve the formulation of the blood sample in its suitable dosage form, if it is tablet or capsule or whatever. So the next step is the clinical evaluation. This step or the studies come after pharmaceutical studies. Finally, we end with a new product or new dual product. So we can summarize this step in the following. Of these methods, the following groups: botanical methods, phytochemical methods, biological methods, which include pharmacology, toxicology, and microbiology, pharmaceutical studies, clinical studies as final steps. In this course, we focus on botanical methods, phytochemical methods, and some extent on biological activity, while pharmaceutical and clinical studies fall beyond the scope of this course. The content of this course are divided in four main topics or main titles. The first of them is methodology for analysis of medicinal plants. Under this title, we discuss the macro and micro beside histochemical examination for medicinal plants. Also, a general phytochemical screening and qualitative determination of extract constituents, beside foreign methods, determination of foreign methods that present in plant sample, and the preliminary quantitative determination of active ingredient, beside the chemotaxonomy, which is important for the study of and classification of medicinal plants. The second topic is structural determination of secondary metabolites or structural elucidation, how we can identify or determine the chemical structure of the isolated and purified chemical compound. This involves the study or discussing topics related to the UV, IR, NMR, and MS spectroscopy and their application in determination of the structure of natural products. The third topic is the biosynthesis of secondary metabolites. In this topic, we discuss some reaction and the general importance in secondary metabolites. We can discuss the common reaction involved in the biosynthesis of secondary metabolite in the living cells, if it is whatever it is, plant or animal. Also, the important techniques for the biosynthesis of the secondary metabolite. The fourth topic is the isolation of marine natural products. It is the important area and very active research area nowadays is marine natural products or the products that isolated from marine organism or the organisms that living in water or in oceans or in seas. In this topic, we discuss the general method for collection, isolation of marine natural products. The, objective, the objectives of this course include uh, the following objectives. The first of them is to describe the methodology for analysis of medicinal plant. The second one is to describe the methods for structural determination of secondary metabolites. The third one is to describe the biosynthetic pathway and origin of secondary metabolites. And lastly, to describe the methods for collection and isolation of secondary metabolites. Also, at the end of this presentation, we find the important references to which we can or to which the student can refer for further information concerning the topic of this course.